Today we are here to talk about normal flora versus pathogens. As you know, bacteria is all over us and inside of us to help keep us healthy, protect us from foreign invaders such as other bacteria or fungus, and also help us with daily processes such as digestion. So, what makes something normal flora and what makes something a pathogen? Well, a normal species that is naturally on or inside of you is going to work symbiotically with your body to help you do some type of process. And a lot of times those normal flora, if they're introduced to another location of your body, might end up being a pathogen. So in the clinical micro lab, we go ahead and take specimens that have been collected by clinicians, we culture them and try to distinguish if there is an infection in that certain location, or is what we see on the plate contamination during the collection process. So say if somebody was trying to get a peripheral blood collection or blood culture like what you're looking at right now, if the phlebotomist or the nurse or whoever drew the specimen does not correctly disinfect the skin, we end up having contamination by normal flora. And that's what we're looking at here. If you'll notice, you see what looks very much like staph. So this is actually coagulase negative staph, which the most prominent one on your skin is Staph epidermidis, okay, Staphylococcus epidermidis, and that's what this is, okay. Notice you don't really see any hemolysis here on the blood agar plate. It looks very, very staffy. It's got that, good sized colonies for gram-positive cocci, and it's that gray that is very prominent or very common to staph. So this is actually a contaminated draw, okay, from the skin. So this is normal flora of the skin. Let me get another one. So we're going to move on to a throat culture. Okay, so this one, as you can see, there's a few different organisms on here. Okay, we see the different sizes. We see hemolysis, alpha hemolysis around certain ones and maybe not others. This is a really good representation right here. So we have the greening of the alpha hemolysis, and then we have some other gamma hemolysis around there, which means there isn't any hemolysis. Alpha hemolysis is going to be the partial digestion of the RBC, whereas the gamma means that there isn't any. So this is actually the normal flora of the mouth and the throat. Okay, the green one that you're seeing most prominently here is going to be your Streptococcus Pneumoniae. Okay. All right, we're going to get another one. This one is a urine, okay, and when we think about urines, we do have microflora that's at the close to the opening of the urethra, which is basically the section before the urine actually is out of the body. Okay, so when we talk about UTIs, that's where they're going to start. UTIs are urinary tract infections, and they're due to the inoculation of the urethra by other types of bacteria or yeast from some process that introduces something that's normally not supposed to be there into there. So the most common way is through sexual intercourse, And also if a female wipes the wrong way instead of front to back, wiping from back to front would introduce fecal flora into the urethra. Sex can also do that as well. And or flora from the mouth, depending on the type of actions that are happening there. But what we're looking at here is not pathogenic, okay? When we think about a normal UTI, we're thinking about that fecal flora that may be introduced, and those flora are usually gram-negative rods, such as the family Enterobacteriaceae, and they would grow on this McConkey plate. Notice there isn't any growth on this McConkey plate, okay? And this signifies that this bacteria is not gram-negative. This is actually normal skin flora. It looks very staffy, doesn't it? So if we compared it to the staph we had before, it looks almost exactly the same, okay? There's a little bit of the darkening behind the actual colony, if there's a lot of growth, and that's very similar here. Okay, something that may also cause the UTI would be Enterococcus, and that is not a gram-negative rod, but that's this is not what Enterococcus looks like. Okay, let's move on. Now we're going to do another urine culture. I have to open it, hold on. Okay, this one is also another urine culture. It was inoculated on both the blood and the McConkey, but notice there's nothing on the McConkey, okay? So knowing what we know about the normal flora, we're expecting that this is not necessarily a UTI. And then we look here at the different types of organisms that are present here, and we have 3 or more different types of colonies here. So that is indicative of contamination by normal microflora. Okay, and so this is urogenital microflora that is normal and not considered pathogenic. All right, that's a really beautiful picture. Okay, let's move on to another one. This time, we're going to be looking at what pathogens actually look like. Unfortunately, these are not going to be cultures that are from an actual specimen with anything else that might have grown. This is actually purified cultures. So this is a pathogen from, where is it from? It is from a urine. Okay, and just as we had talked about, this is actually Enterococcus Faecalis. Okay, notice that this is not the streaking of a urine culture again, because I ended up getting pure cultures from the actual specimen. So the tech that made these for me was so gracious to do this for me, but I wasn't asking for pure cultures. I wanted to see what it looked like if the pathogen was just, actually inoculated on there with any other microflora that might be there. But I am so thankful that they did this for me. So this is Enterococcus Faecalis, okay, and this is from the gastrointestinal tract, as we just said. But notice it looks, it does look different from the staph that we saw on that other culture. Okay, it is kind of grayish or lacking in color, depending on where exactly you look. Usually strep ends up being a smaller type of colony than staph, and Enterococcus kind of falls in between. I guess that's really all I'm going to say about that, but notice that there isn't any hemolysis here. Enterococcus can have different types of hemolysis, but this strain does not. And that was from the urine culture. It would not have grown on the McConkey, just like when we looked here that there was no growth on the McConkey. Okay, here is another one. This is from a sputum. Okay, ooh, this is so much different than what we've seen before. These colonies are a lot bigger. They spread out, but that's not swarming, okay? Please understand that's not swarming. They definitely spread out more, okay? And this is actually a gram-negative rod. This is going to be Klebsiella Pneumoniae. I think that's what we have, yeah. Klebsiella Pneumoniae, and this is actually antibiotic resistant Klebsiella Pneumoniae. Oh, come on, focus. Notice that it has kind of almost like an apron around these end parts of the lines, okay? It kind of fans out. That's very, very common with a GNR, okay? Your Enterobacteriaceae family ends up having that a lot. And the colonies look bigger because depending on the size of the rods are bigger usually than the cocci, okay? So you would expect larger colonies anyway, but that scalloped edge is very common with the GNRs. Okay, so this is a pathogen from the sputum, all right? And it looks very, very wet and shiny, and you'll find that a lot of respiratory organisms end up having some type of capsule and end up looking shiny and wet. If this had actually been on a plate with the that had just been plated from the specimen, since it's a pathogen, you would notice that it would basically take over the plate. The family Enterobacteriaceae divides very rapidly anyway, but that's basically one of the big reasons or big ways that you can tell if something is a pathogen versus normal flora is the existence of hemolysis. And then also looking at its gram stain and deciphering whether that type of organism would be in that location from which the specimen was collected. This is another purified culture this is from the appendix, okay? So this person had acute appendicitis. I don't mean to sound excited about that, but this is E. coli, Escherichia coli, and it is a GNR as well, and it is part of the family Enterobacteriaceae. You don't really, can't really, there we go. We can see some of that scalloping there. If my phone would focus, I'm so sorry. There we go. This one, I believe, is antibiotic resistant as well. That is something you need to consider also when you're doing testing on different organisms, especially from the Enterobacteriaceae family. When we talk about ESBL, that's extended beta-lactamase, and it's a protein, an enzyme that enables the organism to be resistant to different types of drugs that are beta-lactam drugs. And those drugs, they break the beta-lactam ring that are in the cell wall of the organism, the bacteria, and that's how they kill the organism or stop it from reproducing and spreading an infection. But those types of organisms that do have beta-lactamase end up making it so that the antibiotic is not effective. So when we okay, so this one is from the right thigh. Okay, this person had some type of wound on their right thigh. And this is the pathogen that came out. Okay, so let's look at this. We see that the colonies are much smaller than what we've been seeing. Okay, and there is, let's see if you can see that. There is beta hemolysis. I don't know if you can really pick that up. Let's see, there you go. So there is beta hemolysis. Isn't that gorgeous? All right, and as you can see with the big Sharpie writing through the middle of it, it is group A strep, and that is Streptococcus Pneumoniae. Okay, and that was, I believe, one of the organisms that was on the monster inside of me or whatever that show is, and it's because it has hemolysis and it's able to get into your connective tissue or your skin, you know, and spread. So you definitely need to take care of an infection with group A strep, or else it goes systemic and you can definitely die from it. So let's move on from that. So if that was on a plate, a culture from a specimen, that would take over as well, and it would be beta hemolytic, and you wouldn't expect that to be in your throat, especially since your throat has a mucous membrane. All right, so this one is going to be fun. All right, this one's from the left arm. Look at that. Isn't that gorgeous? Okay, so this, I'm assuming, was from a wound on the arm, and this, as you can tell, is a GNR in regard to that that scalloping, but also there's what's called swarming, and you can see it because it's waves of growth. This organism has covered the entire plate. Okay, notice in another one, if we're looking at a blood agar plate, so the blood agar plate should look like this, and you should see a definite distinction between where the growth is and where the plate is. But this you cannot because it has grown all over the place. Okay, that's what swarming looks like. It just looks like waves of ocean of bacteria. And you should smell what I'm smelling on this other end. When you open up E. coli, this is not E. coli, but when you open up E. coli, it has a distinctive smell. So does Pseudomonas aeruginosa. It smells like grapes. There really isn't a good, I don't really have good verbiage to tell you what E. coli smells like, but bacteria, certain types of bacteria have definitive smells. And I think this is Proteus. Yes, this is Proteus mirabilis. Okay, it's a big, big swarmer. And when we look at wound infections and the primary setup, we end up usually doing a different plates that try to inhibit Enterobacteriaceae family bacteria because some of them swarm, just like this one, Proteus does. And they also are rapid growers, okay? They divide very quickly. This is just gorgeous. I don't really want to take this off, but we've got to move on. So this obviously would take over the plate and just eat its way around the plate, and you could definitely tell that was a pathogen there. All right, this one is from a sputum. There's that distinctive smell. This one only grew or will grow on the chocolate agar plate. So that means it's fastidious and it is a finicky eater. And the main one that we're thinking about with that would then be your Haemophilus. Okay, we don't really have what this is here. It could be influenzae, Haemophilus influenzae, or para-influenzae. But this is what Haemophilus looks like. And if it's influenzae, it's not going to necessarily grow. It's not going to grow on your blood agar plate. They would also do other testing. You've seen it in a different video, the X and the V testing to see what it needs in order to grow, and that helps speciate it. All right. This one is this one is from a butt. I don't know. I think it would be around the anus. I'm assuming it's a swab. It doesn't really say what the type of specimen is, just says it's a buttock. So it could be some type of boil or something on the buttock, but I would think that this is most likely from a pregnant woman. So this is actually group B strep. That's Streptococcus agalactiae. And what we're concerned about with this type of organism is if this was from a pregnant lady, we would be concerned about her giving birth and passing this on to her newborn, because as the baby travels through the birth canal, they are exposed to all different sorts of microflora, and if the mother has this, then it can cause really terrible infections with babies, especially the very especially prevalent one would be meningitis. Okay, so if Strep agalactiae gets into a baby's CSF, then they would have meningitis, and that's very serious, and that could be very fatal. Okay, so notice it's a smaller size, doesn't always have color. That's very distinct of Streptococcus. Doesn't always seem to have color. It's usually small. All right. Now we have pathogen from a stool. Okay, so we see the scalloped edges, so we're assuming that this is a GNR. Okay, and that is correct. This is Salmonella. Okay, and I mean, we've already talked about GNRs and Enterobacteriaceae and all that. So Salmonella can cause a whole ton of infections. Usually it's foodborne or waterborne in regard to eating or drinking contaminated food or water, and it would be causing someone to have a lot of gastrointestinal distress, very uncomfortable, and you end up having diarrhea. It really just depends on what the infection is or what the infection type is. Okay, so that's it. That's what you need to know about pathogens versus normal flora. Let's recap real quick. Normal flora is going to not really have hemolysis. All right, it can be a contamination of specimens depending on whether the culture swab was taken aseptically or not. Okay, and normal flora in a place where it's not supposed to be ends up being a pathogen, and if it is a pathogen, usually they don't always swarm. I just like this plate. They take over the plate in regard to it's the most prominent growth that would be on the plate, and those would be the types of things that you need to work up. But the main thing is that you need to make sure you're remembering what the site of collection was. Okay, if you don't know the site of collection, then obviously you can't really judge anything. But those are the types of things you need to look for, and so therefore you need to know what is normal flora and what are the general characteristics, normal characteristics of those different bacteria. All right, thank you so much for watching. If you enjoyed this video, please subscribe, and I try to put new ones out when I can. Thank you so much for your support, and I'll see you next time. Bye.